we have seen that the contributing factors to enhance the reaction rate is the decrease of activation energy, the specificity of the enzyme, and the induced fit. We have seen also that, surprisingly enough, only few residues are needed for the catalysis. So the question is, so why enzymes are so big compared to the size of their active site? How does the tertiary structure of this enzyme contribute to catalysis? If we take the example of an enzyme called alcohol dehydrogenase, its size is in the 100,000 Dalton, while the size of the substrate, which are ethanol and NAD+, are less than 1,000 Dalton. If we take a closer look on this PyMol simulation at the active site of ADH, where ethanol and NAD+ are bound, we see that there is about 10 residues that surround the pocket where the two substrates are bound. And out of these 10 residues, maybe only 2 or 3 contribute to catalysis. So the question is, what is the function of the rest of the enzyme? Is it to keep structural biologists busy? Probably not. There is, in fact, several reasons to explain the role of the tertiary structure of the enzyme outside of the active site. First, the three-dimensional folding of a long polypeptide chain allows the correct positioning of catalytic residues that will react with the substrate. Then you have the region of the protein that is necessary for the induced fit, for the change of conformation that is needed to optimize the positioning of reacting groups between the substrate and the enzyme. Then you have a need to regulate the activity of the enzymes, because these enzymes contribute to the catalysis of reaction in different metabolic pathways that have to be controlled and coordinated. And the last reason why you have protein that are much bigger than the active site of the enzyme is that there are regions in an enzyme that are involved in protein-protein interaction. And in some cases, enzymes assemble in multifunctional complexes, and that increase the efficacy of catalysis. So for example, enzymes that are involved in the catalysis of sequential reaction can assemble and form complex that will catalyze basically the full biochemical pathway. And we'll see examples of that later in the course. So we can try to figure out with a very simple model why an enzyme cannot be limited to a short peptide that will contain all the catalytic residues. This peptide would have to bind the substrate. However, a short peptide has limited folding possibilities. There is an inflexibility of the length of the bond. There is also somewhat fixed bond angles that prevent a folding that would allow for the binding of the substrate and the correct positioning of reacting groups. In contrast, in a large polypeptide, the folding can easily form an active site where the substrate can bind and can contribute to the correct positioning in space of the reacting groups. So the substrate can bind and catalysis is facilitated. Now we can also see how the tertiary structure of an enzyme contributes to its specificity with a very specific example. We are coming back to our enzyme, an enzyme that we described before, the alcohol dehydrogenase. Alcohol dehydrogenase catalyzes the conversion of ethanol into acetaldehyde. And this reaction is coupled with a reduction of NAD+. So if we just ignore the enzyme spatial organization, we can expect the following. The hydrogen from the hydroxyl group of ethanol can give rise to the proton in our reaction, and the 2 hydrogen carried by the methylene group of ethanol would have an equal probability to be transferred to the molecule of NAD to form NADH. What is in fact observed is that it's always the same hydrogen that is transferred from ethanol to NAD. How can we explain that? It's explained by a model of interaction between substrate and enzyme called a 3-point attachment model. In this model, substrate interact with the enzyme via 3 specific attachment sites. And that will fix the orientation of the substrate in the active site of the enzyme, and will bring a certain number of chemical groups in proximity to other chemical groups, and these are the ones that will react together. So let's take a closer look at the active site of ADH. We can see that ethanol is bound to ADH at 3 different points. The methyl group of ethanol attaches to the enzyme. The hydroxyl group of ethanol is also formed the second point of attachment with the enzyme. And 1 specific hydrogen from the methylene group form the third attachment point with the enzyme. Now, hydrogen from this methylene group is positioned nearby the NAD molecule, and it's the one that will be transferred, that will be always transferred to NAD.
I see why a very short peptide could not fold properly, but why can't correct folding be achieved by a shorter version of the full polypeptide chain?
Well, remember that I mentioned that there were other reasons why the size of the enzyme was much bigger than the size of the active site. And your question gives me a perfect segue to the second half of this video. So regions outside of the enzyme's active site play a critical role in the activity of enzyme. First, some of these regions are needed for the conformational change that occurs when the substrate binds to the enzyme. And to show that, I will take the example of an enzyme that we have seen already, the citrate synthase. So this enzyme catalyzes the transfer of an acetyl group from acetyl coenzyme A to oxaloacetate to form citrate. This is the first reaction of a very important biochemical pathway called the citric acid cycle pathway. The concentration of oxaloacetate is extremely low in the cell. For the reaction to proceed, it requires a very high negative free energy. The molecule of acetyl coenzyme A has a very high hydrolysis potential that drives this exergonic reaction. But that comes with a problem. Because acetyl coenzyme A is highly unstable, has this high hydrolysis potential, there is the risk that the hydrolysis of acetyl coenzyme A will happen before oxaloacetate is bound to the enzyme, and we would have a wasteful hydrolysis of one of the substrates of the reaction. So citrate synthase can solve this problem in a very neat way by introducing 2 sequential induced fit. So the citrate synthase, free of any substrate, cannot bind to acetyl coenzyme A. It can only bind to oxaloacetate. When oxaloacetate is bound to the enzyme, you have the first induced fit that creates the binding site for acetyl coenzyme A. When acetyl coenzyme A is bound, then you have formation of the reaction intermediate called citroyl coenzyme A. This is just the formation of a covalent bond between the acetyl group from acetyl coenzyme A and oxaloacetate. Once this reaction intermediate is formed, you have now a second change of conformation, the second induced fit, that leads to the repositioning of catalytic residues that can now trigger the hydrolysis of the acetyl group and its transfer to oxaloacetate. Finally, citrate and coenzyme A will be released. So now, in this spinal simulation, what you can see is the change of conformation that occurs when oxaloacetate, the first substrate, binds to citrate synthase. If the protein, the enzyme, was a relatively short polypeptide, it will be too short to accommodate this very large change of conformation. So that's why you have proteins that are much bigger than the active site. There are other reasons. The second reason is enzyme regulation. So the activity of enzyme is regulated by different signals in the cell. And an example of a signal that regulates the activity of an enzyme is transient covalent modifications. And the most common one is the addition of a phosphate group catalyzed by a kinase, or the removal of a phosphate group catalyzed by a phosphatase. In eukaryotes, the residues that are phosphorylated or the amino acids that are phosphorylated are serine, threonine, and tyrosine. The phosphorylation or dephosphorylation of these residues can either activate or inhibit an enzyme. We'll take an example, which is the enzyme called PFK2, or phosphofructokinase 2. It's a very important enzyme that catalyzes the conversion of fructose 2,6-bisphosphate and fructose 6-phosphate. And this ratio of these two molecules is important because it determines the fate of glucose molecule in a cell. PFK2 is one protein with two domains. One domain acts as a kinase, the other domain acts as a phosphatase. You could imagine that in this bifunctional enzyme, the two domains with opposing function don't work together at the same time. In fact, what determines the activity of the enzyme is a phosphorylation. So when the kinase domain of PFK2 is phosphorylated on a serine, the kinase domain is inactivated and PFK2 acts as a phosphatase, and it will convert fructose 2,6-bisphosphate into fructose 6-phosphate. In the absence of the phosphate group on the kinase domain of PFK2, the phosphatase domain of PFK2 is inactive, and PFK2 acts as a kinase that will convert fructose 6-phosphate in fructose 2,6-bisphosphate. So you see here that a simple change, a phosphorylation of a dephosphorylation, completely changed the activity of the enzyme from being a kinase to being a phosphatase. There is another important type of regulation that is called allosteric regulation. And allosteric enzymes control very critical steps in many metabolic pathways. You have signal molecules, small molecules, called allosteric effectors that can bind to the allosteric enzyme outside of the active site of the enzyme and modulate its activity. We will discuss in detail allosteric enzyme when we talk about metabolism later in this course. So in conclusion, the reason why you need a protein that is much bigger than its active site is it contributes to the positioning of the reacting groups. It allows for induced fit that optimize catalysis. It's necessary for the regulation of the enzyme, or the activity of enzyme, and it's important for the formation of enzyme complexes.